Hello students, welcome to EPG Pathshala. I am Dr. Jiten Vasist from Department of Biotechnology and Bioinformatics, JP University of Information Technology, Solan, Himachal Pradesh. Today we are going to talk on module Gel-based proteomic analysis using mass spectrometer from paper Biophysical Technique 1. Learning objectives of these modules are prepared for identification and characterization of proteins using different gel technologies coupled with mass spectrometry. The main emphasis is given for protein separation technologies and quantitative proteomics. Information related to gel-free quantitative proteomics would be provided in another module of mass spectrometry section. Before going to start, I want to ask you one question. What is proteomics? Actually, this term proteomics was coined by Mark Wilkins, a student of Australia, Marcus University, and he coined this term proteome in 1994. It is the biological assay or tool for determining a gene function in terms of proteins. Proteomics is the study of protein complement of the genome at a specific time. It bridges the gap between genome sequence and cellular behavior of the cell. It shows the direct characterization of a sample's protein in a mixture of proteins and give the answer of following question, that is, what proteins are present, what isoform of each protein is present, and lastly, how much of each protein is present. Now, question comes that whole genome sequence complete in 2000 but does not show how protein function or biological process occur. Moreover, protein post-translation modification sometimes chemically modified or regulated by other after synthesis. Protein fold in specific 3D structures which determine their functions. This proteomics gain insight into alternative splicing also. It aids in genome annotations. And lastly, it is utilized nowadays also for identification of biomarkers associated with different traits. Protein-protein interactions are crucial component of integrated biology. Protein states the covalent modification, cellular localization, presence of different ligands, alternate splicing, proteolytic cleavage, oligomeric state, and protein conformation. All these can be studied with the help of proteomics. That's why we require proteomics in nowadays in modern biology. This figure shows the comparison of past and present scenario of research using newer technologies of genomics and proteomics. In older days, we required more time to analyze the expression of all genes, for example, cancer expression. While in these days, newer technologies of proteomics and genomics saving our time, and we can see the global expression even in a single experiment. In gel-based proteomics, proteins are resolved on gels, preferably polyacrylamide gels, and thereafter these are identified using mass spectrometry. There are multiple modifications in separation technologies and which are according to the need, which is start from one-dimensional gel electrophoresis, that is SDS page, native page, acid urea page, and isoelectric focusing. And later on, people have found the combination of two one-dimensional gel electrophoresis resulting a better resolution. That's we have two-dimensional gel electrophoresis, which is in which proteins are separated on the basis of firstly the pi and lastly they were separated on the basis of molecular mass. Two-dimensional gel electrophoresis. This two-dimensional gel electrophoresis was first introduced in 1975 by O. Farrell. After that time, this technique has re-evolved in 1990s at different levels and become the method of choice for proteome map generation and differential proteomics. In principle, in this technique, proteins are separated on the basis of two characteristics that are charge and the mass. Proteins are first get separated by isoelectric focusing, which is based on their net charge at different pH values. The second dimension is performed on SDS page, and proteins now separated on the basis of their molecular weight. This technique has many-fold resolving power as compared to IEF and SDS page alone. Due to this technique, one can visualize 50 folds more protein than SDS page as protein spots. Moreover, multiple spots containing different molecular forms of the same protein may also be seen on a single 2D. You can compare the different biological samples on two-dimensional gel electrophoresis. You can get a proteome map of healthy person and the diseased person, or you can compare the cancer tissue with a benign tissue or normal sample. Even you can compare the drug-resistant bacteria with drug-susceptible bacteria, and so on. After 2D page, proteins can be detected by using staining, in which we'll find silver staining, Comasi staining, or fluorescent staining. Comasi staining is one of the widely used staining which is inexpensive and it's quick and easy to use. You will find it is one of the common methods, so that there are many published techniques available for this Comasi staining technique. Now you will find there is another technique of staining that we call silver staining. If you compare the previous gel and this gel which is showing in the figure, you will find the number of spots are more, and these are the same sample of the same bacterium, and we have done silver staining. Silver staining is highly sensitive staining. It has varieties of commercially available kits which can be utilized for staining the proteins. It can be easily compatible with mass spectrometry, but this staining has also several disadvantages. Some issues are having with the non-specific bindings. Secondly, it requires good quality substrates for developing the stain. Third is problems with inaccurate relationship between intensity of and protein concentration. And sometimes you will find this technique is labor-intensive procedure. After two-dimensional gel electrophoresis, the gel is developed with the help of silver staining, and we are showing scanner which is scanning the image of silver stained 2D gel. Now coming to the newer gel-based technology that we call difference in gel electrophoresis. DIGE is a gel electrophoresis based technique for protein quantification in a complex mixture. DIGE helps in quantification of differentially expressed proteins as well as those that are common between samples. In this technology, first the proteins are labeled with fluorescent dyes, basically cyanine dyes, this Cy2, Cy3 and Cy5, followed by separation on a single 2D page. The different protein samples are labeled with the different dyes mixed together and finally separated in a same gel, minimizing gel-to-gel variation. Now we will discuss the experimental procedure of differential in gel electrophoresis. If you want to compare the proteins of two samples, that's control sample and the treated one, we can compare on a single gel electrophoresis with the help of differential in gel electrophoresis. Sample number 1 or control sample can be treated or labeled with Cy3 dye. Sample number 2 or the treated sample can be labeled with Cy5, and the pool fraction of both the samples makes pool internal standard that can be labeled with Cy2. All the three dyes are required for labeling three different samples, and these can be mixed after labeling and run on a single 2D gel electrophoresis. After completion of 2D gel electrophoresis, images can be visualized with the help of a floor imager, that we have typhoon imager. This imaging process shows excitation and emission of different dyes at different wavelengths, and because of this you will find three images from a single gel. Size 3 will show the excitation approximately 530 nanometer and the emission is 580 nanometer. Size 5 has an excitation of 633 nanometer and the emission of 670 nanometer. Size 2 has an excitation of 488 nanometer and emission of 520 nanometer. And because of this you will find three gels from a single gel. You can compare the three gels independently, as well as you can overlap all the three images with the help of typhoon imager. And lastly, there is a quantitative analysis which is based on the relative intensity of fluorescing labels at a specific spots, relative quantification or label standards. This can be done with the help of decider software, which is coupled with this typhoon imager. Now we will show you the result output of dyes. For this matter, we'll show you the result which are published in a research journal. In this experiment, the outer membrane fraction of a gram-negative bacteria, Acinetobacter baumannii, has been taken, and the comparison was done on these membrane fraction of native strain, which is ATCC 19606, and the resistant strain, and the two-dimensional electrophoresis was done on 13 cm gel strip with a pH range of 4 to 7. Panel A is showing Psi3 labeling with ATCC outer membrane proteins. Panel B is showing Psi5 labeling with outer membrane proteins of resistant bacteria. Panel C shows the pooled fraction of both outer membrane proteins labeled with Psi2 dyes. The gel image showing the overlap image of all the three dyes labeled proteins, and from this you can see which are the proteins which are differentially expressed and the proteins which are common in both. These images were further analyzed using decider software. Shows the representation of comparative decider gel analysis of normalized gel of native ATCC strain and the resistant strain using BVA module. All the combined results are displayed for a particular spot, that's spot 6. Panel A shows the spot 6 in both the compared strain. Panel B represents the graphical representation for all the three independent experiments. Panel C shows the mean value of 3-day expression, while panel D shows the statistical measurement of these spots. Shows the experimental workflow for proteomic analysis using dyes technology. I'm just showing a comparative dyes analysis which was previously discussed. For that matter, we require protein labeling, the first thing, then pooling of all three experiments, then third is 2D dyes. After that, image is acquired in separate channels, which is followed by image analysis and data quantification with the help of decider. The protein spots on the basis of differential expression will be chosen, and then they will further followed for mass spectrometry analysis, which may be followed after that for protein identification using bioinformatics technologies. If you are not using dyes, the same strategy can be employed for 2D gel electrophoresis or even 1D gel electrophoresis, that's SDS page. For that matter, firstly, the fractionation and isolation is done on 2D. Then proteins of interest will be taken out and they are get trypsinized into peptides. And lastly, they go for mass spectrometry identification and database searches for protein identification. It means You will require mass spectrometry also for identification of proteins, and these proteins are sorted on the basis of mass by charge ratio for mass spectrometry. That is m by jet ratio. M is the molecular weight divided by charge on this protein. This figure shows the generalized protein identification by mass spectrometry. For this, we require proteins or the spores which are removed from gel. They are further degraded or fragmented using trypsin or any endopeptidase, and there is a spectrum is generated on mass spectrometer, which is next matched with the databases which are present in the bioinformatics tools. For that requirement, we require the databases which are artificially built up with the help of bioinformatics tool, and the library is created. So there is a comparison between the fragments which are generated by experimental procedure and the fragments which are generated with the help of artificially trypsinized proteins. This slide shows that proteins can be digested with the help of proteases, which are specific for cleaving protein at specific site. For example, trypsin can digest protein if it will find lysine or arginine, and their C terminal will be digested with the help of trypsin. You can predict the peptide cleavages with the help of different enzymes. Peptide cleavage can be done with the help of tools which are bioinformatic tools available on free on XPSI sites. You will find there are many enzymes which can cleave proteins at specific sites. However, trypsin is one of the enzymes which is widely used for proteomic analysis. There are certain advantages of trypsin over other enzymes. It is a robust and stable enzyme. It works over a range of pH values and temperature. It is quite specific and consistent in cleavage. It cuts frequently to produce ideal molecular weight peptide. Sometimes you will find it's inexpensive and easily available. It also produces some autolysis peaks which may be utilized for calibration of mass spectrometer. This trypsin is very specific. You'll find that it can cleave arginine and lysine. It's very active, and you will find that sometimes it's resistant to digest itself also. Peptide mass fingerprinting or PMF, which has a similar principle as of our fingerprints, that the fingerprints are generally having a specific fragmentation of individual person. The same thing applies in peptide mass fingerprinting. It depends on the fact that if a protein or peptide is cut up or fragmented in a known way, the resulting fragments or resulting masses are unique enough to identify the protein. This technology requires a database of known sequences. It uses software to compare observed masses with the masses calculated from database. It shows the result of peptide mass fingerprinting as a spectrum which has came from a purified protein of a bacteria and digested with trypsin. You will find the different databases which are related to peptide mass fingerprinting. The most common are Profound, Mouse, Peptide Search, Peptident, and Mascot. Mascot is one of the widely utilized database which is available on net for peptide mass fingerprinting. This slide shows the comparison between their theoretical mass and the experimental mass which has came from experiment. The theoretical mass has came from bioinformatic tools, while experimental data can be compared with theoretical mass. Now, coming to the advantages of peptide mass fingerprinting. Actually, this peptide mass fingerprinting shows inexpensive form of mass spectrometer, and it's very robust. It does not require too much sample optimization, and it can be done by a moderate skill operator. It does not require a MS expert person. It is widely supported by different web servers, and it improves a lot as databases get larger and instrumentation get better. And moreover, one can see that it is high throughput robotics can be utilized, and we will go for 500 samples a day by this peptide mass fingerprinting. Now, this peptide mass fingerprinting has also some drawbacks. The major limitation or the drawbacks are that it requires that the protein of interest already be been sequenced in a sequence database. If it is not there, then it will be a problematic. Moreover, missing critical mass peaks always lead to a problem. Thirdly, the mass resolution or accuracy is very critical. Best to have less than equal to 20 ppm mass resolution. And lastly, it is generally found to be only about 40% effective in positively identifying gel spots. Tandem mass spectrometry. Tandem mass spectrometry has a purpose to fragment ions from parent ion to provide structural information about a molecule. It allows mass separation and amino acid identification of compound in a complex mixture. It uses two or more mass analyzers which are separated by a collision cell filled with argon or xenon gas. Collision cell is where selected ions are sent for further fragmentation. Shows the experiment which is based on MSMS for proteomic analysis. In the first, the proteins are digested and they are further go for MS-based peak identification by peptide mass fingerprinting. Then the second collision cell can be utilized for tandem mass spectrometry, and you will get a MSMS spectrum which is further utilized for protein identification using bioinformatics tool. Now coming to the application of proteomics. Proteomics has a wide applications in medicine, in plant research, and in other several ways of toxicology as well. First, number one, the clinical application of proteomics, that people have found the different biomarkers associated with different diseases. This proteomics has also a direct relation with the drug development and the discovery programs. There is a pharmacoproteomics is also there. And lastly, to the research level, people have found protein-protein interactions, even the membrane-protein interaction has been found as an application of proteomics. These are several future applications which can be utilized in medical sciences with the help of proteomic analysis. Proteome level studies will be used increasingly both to identify and follow the course of various pathological conditions. This technology can allow an improved understanding of the mechanism of action of many of the drugs that are routinely administered, and also the effect of these therapeutic drugs on protein expression. In addition, proteomic studies will increasingly be used for both diagnostic and prognostic purposes in several diseases. Although there are so many advantages of proteomics, but this technology has also led with some limitations. The limitations are solubility of individual proteins differs, and because of this you will find different reproducibility problems associated with 2D dyes or 2D experiments. Secondly, 2D gels unable to resolve all proteins in a given time. Most proteins are not abundant like kinases, so their expression levels are different and these cannot be detected several times. Protein not in database cannot be identified using proteomics. Multiple run can be very expensive. Proteins are fragile and sometimes can be degraded easily. Proteins exist in multiple isoforms and sometimes these isoforms are not even identified. No protein equivalent of PCR exists for amplification of small sample. So students, let's now summarize what we have learned in this module. The whole complement of proteins of an organism or a cell at a specific time is termed as proteome. This is the first thing what we have learned. The term proteome was firstly coined by Mark Wilkins in 1994 as an analog of genome. Proteomics is the complete study of all proteins, their abundances, variations, and modification of a cell or the organism. If you see that proteomics has several advantages over genomics because gene transcript cannot exactly tell us about the abundances of a particular protein within a cell due to the alternative splicing, mRNA degradation rate, and post-translation modification of proteins of a cell under different circumstances. In gel-based proteomics, proteins are dissolved on gels, preferably polyacrylamide gels, and thereafter identified using mass spectrometry. There are multiple modifications in these biochemical separation methods according to the need. For example, one-dimensional gel electrophoresis, that is comprises of SDS page, native page, aciduria page, or isoelectric focusing, and two-dimensional polycarbonate gel electrophoresis, basically blue native pages, 2D difference in gel electrophoresis, or 2D dyes. In 2D gel electrophoresis, proteins first separated by isoelectric focusing, which is based on net charges at different pH values. The second dimension is performed on SDS page and the proteins now separated on the basis of their molecular weight. This technique has manifold resolving power as compared to the isoelectric focusing or SDS page alone. Okay, the proteomics is not only limited to high throughput identification of proteins. However, because of newer methodologies and advancement in identification technologies of mass spectrometry, it can be utilized for prognostic and diagnostic disease biomarker discoveries, protein-protein interactions, protein network identification to understand cellular processes. If you see dyes help in identification of differentially expressed protein as well as those that are common between two samples. First, the proteins are labeled with fluorescent dyes. Basically, they are cyanin dyes, site 2, site 3 and site 5, followed by separation by 2D phase. The different protein samples are labeled with different dyes mixed together and finally separated in a single gel, which minimizes gel to gel variation. After dyes or 2D gel electrophoresis, proteins of interest are identified and then they are proceeded towards mass spectrometry identification. In the mass spectrometry identification, first thing comes which is peptide mass fingerprinting. As the name suggested, this peptide mass fingerprinting depends on the fact that if a peptide or a protein is cut up or fragmented in a known way, the resulting fragments or the resulting masses are unique enough to identify this protein. This is done with the help of different enzymes. Basically, these enzymes are the like trypsin, which cleaves the proteins at a specific points, and after this proteins are separated on mass spectrometer by peptide mass fingerprinting. And later on the newer technology has came out that we call the tandem mass spectrometry, which allows mass separation an amino acid identification of a compound in a complex mixture. It uses two or more mass analyzers which are coupled together, or in between these are separated by a collision cell filled with argon or xenon gases. This collision cell is required for refragmentation of refragmentation of masses, and because of this you will find the amino acid identification. This is the workflow by which we'll get a protein identification start from our SDS page analysis or our 2D page analysis, and lastly it will reach up to the level of mass spectrometry. But there are certain limitations are all associated with gel based proteomics. First limitation, which is found in several ways, that's the solubility problem of the proteins, that certain proteins cannot be solubilized properly, so you will get lesser number of proteins if you see in gel based proteomics or gel free proteomics. Second thing is that some proteins are not abundant like kinases, so if the expression is lesser, there is a lesser chances of identification of these proteins. Moreover, proteins which are not in database are very difficult to be identified or sometimes cannot be identified. Moreover, multiple runs require more money and more expensive. And lastly, you will find the proteins are the fragile molecules and they can be degraded easily, and most of the time you will find the reproducibility problem comes because of this gel based proteomics. Thank you very much.